Okay, good evening everyone. Good evening, good evening. Good evening every one of you. Good evening. Okay, let's wait for a minute. Let others also join. By that I will tell how to study. We will study in a very simple way. Okay. Okay. Good evening, good evening. I'm good, Sneha. Thank you. Okay. So friends, every one of you listen to me very carefully. How to study right now I'm going to tell you. Just follow that very correctly without having any kind of, confusions. Just listen to me correctly. Okay, since it's a first class and we have finished only two hormones and we have to finish the gibberellin hormone, right? So what we are going to study? We are studying 40 to 45 minutes. Listen every one of you very correctly how I will study and what exactly to study. Okay, so in this particular class, everyone follow my words correctly. First, listen to me carefully. We have to finish the hormone auxin. We have to finish the hormone gibberellin. Okay, so we don't spend much time on these. So since it's only two hormones in the tomorrow class, we have to revise all the hormones. So it will take much time. So since only two hormones, we don't keep much time at all. We will keep minimum 20 minutes and maximum 25 minutes for the each hormone. Okay, don't study beyond that level. Okay, how to study? Okay, I really don't know what kind of book you are following. Okay, so what you have to do, first keep the PPT with you. I hope everyone is having our PPT, whomever enrolled, keep the PPT with you. After keeping the PPT with you, you can keep one book. Either you can keep the Pathfinder or you can keep the Arihant book or whatever the study material that you are having. Suppose example, if you don't have the Pathfinder, if you don't have the what we can say that Arihant, even you can go for the Lincoln and Tay's book also. Okay, so now see how I will study. See, I have taken the book of Arihant. Okay, now in this the hormone auxin as well as the hormone gibberellin was written in 4 pages, which contain a crystal clear information, more than enough. Beyond that there will be a no way that the question will appear, because as I already told you, from every hormone we are going to get the question very direct and very straightforward question. So now listen how I am saying. So first, while studying the hormones, first every one of you, whatever the book it may be, see, remember I will tell the few contents. First, read the definition of the hormones, then read the properties of the hormones, then start with the hormone auxin. Everyone remember, the moment you start with the hormone of auxin, whichever the book that you are having, please read about the discovery of the auxin. First thing every student has to remember, only read in this order. Don't read anything extra, don't read anything unnecessary. After that, read the types. After discovery, read the types of auxin. After types of auxin, just study the biosynthetic pathway. Study the biosynthetic pathway. And friends, when you are studying the biosynthetic pathway, I want every student has to study only one pathway, that is the IPA pathway. You people no need to study IAN pathway, IAM pathway and TAM pathway. No need to study at all. So study the discovery, study the types of auxins, study the biosynthetic pathway. Then the most important, friends, let's study the inhibitors of the pathway. Okay, which is extremely important. Then final, the fifth one, only study the functions. Okay, then the sixth one, friends, study the signaling. Okay, now remember my words. Apart from this, you people don't study even single point also extra. Okay, I am giving you 100% assurance that the question never ever go beyond that level. Once you study the question, I will ask a few questions. Once I will give the time exactly 20 minutes from now onwards. 20 to 22 minutes will be the maximum. Even I will be start studying with you. I will start studying about the hormones, as well as especially I will start studying about the one hormone auxin. So you know, right, in the textbook or anything which contain extra amount of information. So what you people do, don't study anything information which is present extra. What you do is, whichever the information present in the slide, that information only you read in the book, whatever the book you have. If you have a pathfinder, that is very good. If you don't have the pathfinder, whatever the book that you are having, please start reading that. Only follow this order: discovery, types of auxin, biosynthetic pathway, inhibitors of pathway, functions and signaling. And I forgot to tell one more point, friends. Chemiosmotic model. In the morning class, I did not explain it to all of you. Chemiosmotic model. I have explained, but I did not show you the diagram. Okay. Chemiosmotic model. Everyone has to read only in this order. Now friends, the time is 7:05 p.m. Okay. Now I will give the time up to 7:30 p.m. Okay. By 7:30 p.m. we all has to finish the hormone auxin. Okay. And what you people has to do, if you get any kind of a doubt in that particular line or any particular statement you did not understand, immediately ask me. I will solve it instantly. Okay. So after studying that, we will solve the questions. Okay. So now time is 7:05. Please start studying 7:30 up to 7:30 p.m. Just 20 to 25 minutes. Only one single hormone. I want you to study in this particular order. Okay, let's start studying. Okay, so I am having my book, so I will be start studying by marking. So when I mark, friends, I can remember the concepts very long time. Okay, see, because you know, I strongly believe whatever the receptors present on the fingers, they always carry the impulse till the central nervous system. By that we can remember the concepts very easily. Okay, okay, everyone start right now. So once again, 7:30 p.m. we will talk. If anyone having doubt meanwhile, please type your doubt immediately. I will solve it. I'm good, Saima. Please continue. Yeah. Okay, start. And friends, 7:30, in 5 minutes I will summarize. In exactly 5 minutes I will summarize what I have studied. Okay. And I will ask the question. If you answered that particular question correctly, I believe that you read the concept very clearly. Okay. Now the time starts now. Okay. We will once again talk at 7:30 p.m. Okay. Please start. Any doubt, any statement, if you feel confused, ask me very immediately. See, this is the one which will check our patience. Let's see how many people will study consistently and how many people will leave. Let's see. Every line you study, every point you study. Keep the PPT with you and only read the information present in the PPT. Trust my words, if the question goes beyond that, you come and ask me, sir, the question gone beyond the level whatever we discussed. It never go. Just read it. Study consistently. I will ask total 5 to 6 questions from the hormones. If you answer 5 to 6 questions, your preparation is really good. Be consistent in your preparation. Study with interest. Don't worry about others. Please don't study the tryptophan independent pathway, friends. So only study the tryptophan dependent pathway. There are two pathways are there, tryptophan dependent as well as the tryptophan independent. So especially what is the tryptophan dependent pathway? That is the one which is the IPA pathway. Only focus on this pathway. No need to study the tryptophan independent pathways at all. Okay. Be sincere, don't worry about others, just study on your own like how you are studying at home. Any kind of a doubt, please ask me immediately. Any statement if you feel difficult, if you did not understand. Anyone if did not understand the chemiosmotic model, ask me. Good evening, Amar. Please continue. We are doing the self-study. See, I strongly believe the people who are studying consistently, the people who just come and pass away, they won't be serious, okay? However, you are studying at your home, feel the same environment and study sincerely. Archana, Shivani, are you very clear with this chemiosmotic model? Abdul, Tarun, if anyone feels difficult in the chemiosmotic model, ask me, I will explain. Read every line, understand every line. Oh, sorry. Okay. Sure, Krishna. Please look into this auxin influx and efflux. Okay. Please, everyone else, please continue it without confusion. You people continue it. Whomever having doubt, only look into this. Krishna, please look into this. Auxin actually exhibited in the plant body two types. One we call it as IAAH form, and auxin exhibited in the IAA minus form. When I use a word auxin exhibited in the IAAH form, I can call it as uncharged form. Okay, when I say auxin is exhibiting in the IAA minus form, friends, what is the statement I can tell you? I can tell you that auxin is exhibiting in the charged form. Krishna, remember, if IAAH, uncharged form, if the uncharged form wants to enter into the plant cell, it will enter into the plant cell very directly. There is no need of any kind of a carriers. But friends, if the IAA minus which is the charged one, if it wants to enter into the plant body, compulsory it require two type of carriers. If the IAA minus want to enter inside from one cell to the another cell, it require influx carrier. Please look into this, Krishna. This is the influx carrier. So this is the IAA minus. IAA minus is coming via influx carrier. Now this IAA minus, if it wants to go from one cell to the another cell, so you look into this, they will travel via efflux carrier. Okay, efflux carriers, we can also call it as what proteins, you know, we also call it as the pin proteins. We also called as pin proteins. And in the morning class I have told to all of you polar transport. Can anyone tell me why this polar on what factor the polar transport is depending on? Listen my statements, everyone. The polar transport is exclusively depending on the direction of the pin protein, nothing but the direction of the efflux carriers. What I mean to say, friends, see, suppose example, this is one efflux carrier. Okay, so auxin will be moving like this only. Suppose example, friends, efflux carriers are located in this direction. Now friends, then auxin also move in this direction only. Don't get confused. So what my statement here is, 99.99% of the plant cells, the efflux carriers or pin proteins, they are present at the basal position. Basal positions means at the end of the cell. Since they are present at the end of the cell, auxin is also traveling via end of the cell only, like this only, exact unidirectional transport. This transport only we can call polar transport. Okay, you got it, Krishna? Okay, everyone, please continue, please continue. 8 more minutes are there, not more than that we have to study. Let me know, Krishna, if you did not understand. Very, very good question, Archana. Archana, listen. IAH, IAAH. Correct, na? You look into this. Your question is that why IAH become deprotonated in the cell wall space? You know, no, in the cell wall space, what kind of pH it will be there? Mostly a kind of a neutral pH. Correct, na? So in this neutral pH, which is the predominant form, in this particular neutral pH, IAAH lose this proton. Upon losing the proton, it will become IAA minus, which is the predominant form. Keep it in your mind, Archana, in the cell wall space, IAAH cannot become a predominant form. Only IAA minus, that is the charged one only, can become the predominant one. Uncharged one cannot be the predominant one. So that is the reason only charged one can sustain. IAAH get immediately dissociated in the cell wall space. Got it? IAAH is a non-charged. IAA minus is the charged. Right? Yeah. Okay. Please continue. Please continue. Okay. 7 minutes more. Good doubt. Good doubt. Yes, absolutely, Saima. You look into the picture very carefully. You can look into this. On the cell wall membrane, what kind of pumps are present? H plus ATPase pumps are present. Correct now? What this H plus ATPase pumps are doing? By using the ATP, they are constantly pumping the protons. Once they are constantly pumping the protons, what will happen? The cell wall space become obviously acidic. Absolutely true, because of the high amount of H plus ATPase pumps. And we remember, no, all these H plus ATPase pumps are going to be activated by the presence of IAA only. That is true. 4 minutes more. Wonderful, wonderful. Study with patience. Study with interest. Yes, of course. Correct only, na. In the cytosol, what kind of a pH? Cytosol is a neutral pH, right? So what happen? IAH lose the proton, then it is converted into the charged form IAA minus. So the deprotonation is occurring in the cytosol. Absolutely true. You understand, Osaima? Obviously it will be deprotonated. Lose the proton. 2 minutes more. Okay, friends, now stop. Now stop. Okay, enough of studying. Okay, I hope everyone studied it in a order. Okay, now let me check whether what information you have studied. Okay, what we people will think that whatever the information that we have studied, the question will always appear from that particular part only. Okay, question will come from that part only in a little trickiest way. Okay. Okay, Archana, I will tell that. Just hold on. Just wait a minute. Okay, now every student start answering question. I am going to ask very simple questions, very simple 5 questions. Whether you studied it or not, I want to know. So if you answer these questions, I believe that you can answer the PYQs also very easily for the part B. Every student answer my first question. Among these 6, which are natural auxins? My first question is, among these 6, which are natural auxins? IAA, IBA, 4-chloroindole 3-acetic acid. Number 4 is 2,4-dichlorophenoxyacetic acid. Number 5 is 2,4,5-trichlorophenoxyacetic acid. Number 6 is dicamba. Slowly, slowly, slowly. Every student. What about others? Can anyone tell me? Okay, every student has given the answer IAA and IBA. Okay, so friends, whatever you said is absolutely true. There is no doubt about it. But I want you to understand, friends. Remember, IAA, every student follow my words. IAA is the type of auxin which is present. Everyone follow me first. IAA is the one which is present naturally in all the plants. Every single plant IAA is present. Friends, if someone said 3, okay, so IBA is a natural auxin. It is abundantly present in the corn as well as it is present in the mustard. Okay, but friends, I want every student has to remember 4-chloroindole 3-acetic acid. This is also a natural auxin, friends, which is present in the pea plant. Okay, which is present in the pea plant. Please don't get confused with that and don't forget. Okay, once again, in the morning class also I told to all of you, the most predominant form of the auxin is obviously IAA. We are calling it as a most predominant form. It is present in all the plants. But IBA is also natural, but it is not present in all the plants. It is present abundantly in the corn and mustard. What about this 4-chloroindole 3-acetic acid? It is naturally produced in the plant body. Friends, please remember my words. It is only present in the pea plant. So in a given question, the first 3 are the natural auxins. Next 3 are the synthetic auxins. Okay. Now every student has to answer my question. In this, there are 3 synthetic auxins are there. 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid and dicamba. Friends, dicamba nothing but it is a dichlorobenzoic acid. What is that? Dichlorobenzoic acid only we can call dicamba. These 3 can act like a synthetic auxins. My friends, every student remember, all the synthetic auxins can act as herbicides. What, my friends? All the synthetic auxins can act as a herbicide. None of the natural auxin never ever act as the herbicides. Please don't get confused with this particular point. Okay. Now my second question to all of you. I will ask a very simple questions only. Okay. Now my second question to all of you. I am giving a different type of inhibitor. NPA, naphthyl thalamic acid, TBA, triiodobenzoic acid, which we have studied in the morning class. NOA, naphthoxyacetic acid. I am adding some more. There is another inhibitor called as quercetin. Okay, friends, sometimes they don't use the word quercetin. Instead of using the word quercetin, they use the word flavonol. Okay, so you people don't think that flavonol is not an inhibitor. It is also an inhibitor. Okay, then another one we can call the genstin. Okay, I have given 5 inhibitors here. Now my question is, you all people commonly know that all these inhibitors function is what? Blocking the auxin transport. Yes or no, friends? Either they can block the auxin influx also, or they can block the auxin efflux also. I am not asking what they are blocking. Whether they are blocking the influx or whether they are blocking the efflux doesn't matter to me. My question is, among these 5, which are present in the plant body and block the oxygen transport? Which means my question is very clear. Which are natural oxygen transport inhibitors? Which are natural oxygen transport inhibitors? That is my question. Which are natural oxygen transport inhibitors? Even though you don't know the answer, give the guess. Give the guess. Someone said D and E. Ravi, what do you mean by that? And Ravi, what is my question? You are saying that your answer is that naturally occurring oxygen transport inhibitors are quercetin and genistein. Is it? That is your question. That is your answer. Okay. Mahesh said 134. Okay. Anyone else? We have studied this in the morning. But the only thing is I did not tell the name. They are not a difficult question. 1, 2, 3, 4. T-bond, NPA. What are those? Okay. Quercetin and genistein. Wonderful, wonderful, wonderful. Last 2 are natural. Fantastic. Quercetin and genistein. These 2 are said to be the natural oxygen transporter, natural oxygen transport inhibitors, because they are actually present in the plant body only. Whereas friends, NPA, TBA and NOA, what is the function of in the morning class also we have studied, right? TBA, NPA as well as NOA. These 3 are what? These 3 are the synthetic inhibitors. Very good. These 3 are the synthetic inhibitors, and only quercetin and genistein are the, I mean to say, major, what we can call the natural oxygen transport inhibitors. Wonderful, wonderful. So slowly analyze the question. Don't be confused. Okay. Now, friends, my third question. Okay. Now, I'm giving the 3 processes. Now tell me, oxygen will follow the processes or not? Phototropism, gravitropism and thigmotropism. Thigmotropism. I hope every student know very well what is thigmotropism. Formation of a tendrils like structure, coiling like structures. Now my question is, among these 3 steps, how many steps are followed by the auxin hormone? Among the phototropism, gravitropism and thigmotropism, how many processes can be followed by the hormone auxin? All. Okay. Anyone else? One and 2. Only first 2. Okay. It's a little logical question. You have to think it correctly. Okay. Someone said all 3. Anyone? Okay, friends, actually the 3. Answer will be the 3. Okay. Every student listen my statements very correctly. Okay. It is not a difficult question, but it's a logical question. This is what we can call tendril. Correct na? This is the tendril. Okay. a wire or a coiled like structure. My friends, follow me. You know, right, what is the speciality of a tendril? Tendril always associated with a particular support. With the help of a support, it's keep on forming the coil like structure. Now every student remember, whenever this tendril associated with a particular surface, remember my words correctly, whenever the tendril associated with a particular surface, then only the tendrils starts releasing a little amount of the hormone auxin. But the point is very clear, friends. Hormone concentration is not at all higher. Very low concentration of auxin is going to be released. But overall statement, I can tell you that hormone auxin involved in the thigmotropism. Why? Because whenever the tendrilar structures are associated with the solid support, during that particular time, from that particular tendrils, only from the tendrils, there is a little concentration of auxin is going to be released. If you listen the morning class, I have told you, friends, 90% of the auxin is released in the apical meristems. Shoot apical meristem. I told you 10% or the low concentration of auxin present in all parts of the plant body. So actually the tendrils also consist of a very low concentration of the auxin, not more concentration of the auxins. Okay, so correct answer will be 3. Phototropism, gravitropism, as well as the thigmotropism. But if they ask you the question which of the only 2 major phenomena, whatever you said is absolutely right, gravi and photo. But if they ask which of the 3 processes, so obviously you have to include the thigmo. If they have given the thigmo in the question, please choose the thigmo also. Based on the option, you choose it. Okay, now my next question. What are statoliths? What are statoliths? What are statoliths? What are statoliths? You know, this actually we are going to study in the which concept where we will be studying? We will be studying in the transport of plants. Okay, anyone? What are statoliths? Just now, friends, I have told you very clearly that, friends, auxin will show the gravitropism. Correct, na? Gravitropism is what? Showing the response towards the gravity. So someone has to respond to that auxin. Who will respond? Actually, the gravity sensors will be respond. Correct, na? So I don't know whether you remember my words or not. In the cell biology chapter, I have told to all of you many times. Friends, certain amyloplasts which are present in the roots. Certain amyloplasts which are present in the root. They are acting as the gravity sensors. Don't get confused with that particular point. Very good. So all the students remember the specialized amyloplast. Follow my words. The specialized amyloplast which are acting as the gravity sensor, we will be calling them as the statoliths. Yes, it will act there. Yes, it will act there. Yes. Okay. So remember the gravity sensors, the amyloplast which are present in the root cap, which are acting as a gravity sensor, them only we will be calling it as the statoliths. Very simple point. Okay. Now everyone tell me, what is the name of the auxin receptor? Every student answer my question. Name of the auxin receptor through which auxin will show their action. Name of the auxin receptor through which auxin starts showing their action. Faster, friends. Okay, in the tomorrow class I will finish nearly 4 hormones. Everyone, what is the name of the auxin receptor? Every student has to answer. Through which auxin start acting. Morning class also I have explained it to all of you 3 to 4 times. What is that? It is called as a TIR1 receptor. Everyone forgot, friends. Morning only we studied, right? TIR1 receptor. No. It's not ABC receptors, Ravi. It is called as the TIR1 receptor. Every student answer my question. What is the name? Very good. TIR1 and ABP1 receptors. Very good. Okay, but every student remember, this is also valid question. Remember, what is the difference between the TIR1 and ABP1 receptors? All the students listen my words without confusion. When the auxin uses the ABP1 receptor is whenever they want to synthesize or activate the H plus ATPase pumps. Friends, please don't confuse, okay? Whenever the auxin want to activate or synthesize the H plus ATPase pumps, then only, my friends, auxin is going to do this. Very simple point it is. Now, whenever auxin is going to show the signaling, obviously auxin will utilize the TIR1 receptor. So I can tell to all of you, TIR1 is the major receptor for the auxin, which is asked in the 2020 November also. Now every student answer, what is the name of the, don't confuse my words, don't confuse with my question and answer. What is the name of inhibitor gene? What is the name of inhibitor gene that is blocking the ARF of auxin? What is ARF? We discussed morning class. Auxin response factor. Now my question is, what is the name of the inhibitor gene? Sometimes instead of using the word inhibitor gene, they will also use the word early gene. What's the name of the inhibitory gene or the early gene that is blocking the ARF? I want everyone has to answer this. What is the name of the inhibitory gene? We can also call it as early gene. Wonderful, Mauli, very good. So what do we call them as? Auxin IAA, right? This is what exactly, friends, we will be calling it as the inhibitory gene, or our friends, early gene. Wonderful, 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 wonderful. Okay, so friends, I am 1000% sure beyond this level we never get any kind of a question. Okay, now last and final very simple question. What is the, everyone answer my question, what is the name of the other name for the efflux carrier? What is the other name for the efflux carrier? Other name for the efflux carrier, everyone. This is important, friends. This is very, very important one. Very good. So everyone said that the other name of the efflux carrier is called as pin protein. Now my question to all of you friends, listen my question correctly. In a given 4-marker question, they have asked this particular statement. How many number of times the pin protein transpan the plasma membrane? This is my question. My question is, how many times the pin protein transpan the plasma membrane? Saima, you remember? Sumaya, Deepika, you remember? How many times the pin protein transpan the membrane? Can anyone tell me how many times? You know, right, we will use the word GPCR is the one which transpan the plasma membrane 7 times. And we can say that glycoporins are the one which can transpan the membrane only 1 time. Wonderful, Karan. Fantastic. Very, very good. Pin proteins. Fantastic. Very good, Shivani. Pin proteins are said to be 10 transpan membrane protein. Sumaya, it is 10. Okay. So it will transpan the plasma membrane 10 times with the help of a hydrophobic amino acid. Wonderful, friends. Wonderful, wonderful. Okay. 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 Now you can ask me, if you don't, if we are getting the question beyond the level whatever you studied, trust me, the question never come from that level. Question always come from the level whatever we have studied. Okay. Now everyone answer my question. I am writing 2 types. One is called basipetal, another one is called as acropetal. Okay, basipetal transport, acropetal transport. Everyone answer my question. Among these 2 type of a transports, auxin will follow what kind of, among these two, which is called as polar transport, which will follow the polar transport? Does the basipetal transport will follow the polar transport, or the acropetal will follow the polar transport? Everyone. Does the basipetal will follow the polar transport or the acropetal will follow the polar transport? That's my question. We have studied in the morning class, right? Wonderful, wonderful, wonderful. Which is the basipetal? There is no doubt about it. Which is what is basipetal, friends? From shoot to the root. This kind of a transport only we will be generally calling it as what, friends? This one. Okay. But I am requesting every student remember one simple point. What is the simple point? Very good. Sumaya said both. Okay. Very good. Remember my words. Whatever most of the students said is right, basipetal. Mostly polar transport occur basipetal only. That is from shoot to root. But what my point I want you to remember, in case of a xylem parenchyma, okay, the hormone auxin will follow the acropetal transport. Only in case of here only. Only in the xylem parenchyma, the hormone auxin will follow the acropetal. 99.99% always hormone auxin travel from shoot to root only via basipetal transport only. There is no doubt about it. Right. So don't get confused with that. Okay. Now last and final question. What is the name of the proteasomal complex? What is the name of the proteasomal complex that will degrade auxin IAA gene? What is the name of the proteasomal complex? This is also asked 4-marker question, one of the statement. What is the name of the proteasomal complex that can degrade the auxin IAA gene? What is the name of the proteasomal complex which is degrading the auxin IAA? Very, very good. What do we call them as? 26 proteasomal complex. By what, friends? By the process of ubiquitinylation. Correct, na? Of course, once the auxin and IAA associated with the ubiquitin molecule only, friends, then only the process of degradation will occur. Otherwise, friends, the process of degradation never ever occur. Okay. Okay. Now I hope every student remember all the points without confusing. Okay. Now answer my last and final question. I have told to all of you, auxin promotes apical dominance. Okay, which is inhibiting the growth of lateral buds. What is the hormone that is acting antagonistic to the hormone auxin? What is acting? Yes. What is the hormone that is acting antagonistic to the auxin in terms of the apical dominance? With respect to the apical dominance. Very good. What is that particular hormone, friends? Obviously that hormone is the, what is that? Cytokinin. Wonderful, wonderful, wonderful. Okay. Okay. Okay, wonderful. Okay. So friends, I am giving you a last and final question from the auxin. Last and final question. I want you to think and answer. Okay. So column 1, column 2. Okay. Tomorrow I can't discuss this many number of questions because tomorrow I will discuss the previous year questions first. But I will we will study the all remaining 4 hormones by tomorrow itself. Okay. So option A. Slowly analyze the question, friends. Option A, root differentiation. Option B, vegetative bud differentiation. Option B is vegetative bud differentiation. Option C, flower bud differentiation. Option C, flower bud differentiation. Take your time, take your time. Option D, fruit development. Option C, fruit development. Okay. Now friends, I will give the opposite ones. Okay. Remember the option 1, kinins. Kinins followed by auxins. Use the brain. With a common sense you can answer this question. Kinins followed by the auxins. Okay. Then the point 2, auxins followed by kinins. First option is kinins followed by the auxin. Second option is auxins followed by the kinins. And friends, the third and most important option is auxin plus gibberellins. Option 4 is auxin plus carbohydrates. Auxin plus carbohydrates. Okay, this is the given question. Okay, I think my picture is oh, 1 second. Okay, I will reduce my picture size by that you can see the question correctly. Okay, yes. Look into the question. Yeah, so this will be the question. Now I am giving the options here. I want every student has to answer this question. Okay, take your time, take 1 minute of time, no problem. A, B, C, D. Okay, option A 4, 3, 1, 2. Okay, option B, take your time, no problem. Option B 4, 1, 2, 3. Option C 3, 2, 1, 4. Okay, make sure that this question only came in the exam. Remember that this question appeared in the exam. You have to solve it correctly. I am writing the option D here. 1, 4, 3, 2. Option D is 1, 4, 3, 2. This is the last and final question. You have to answer correctly. I will be reading the question once again, my friends. Follow me. In the column 1, option A, root differentiation. Option B, vegetative bud differentiation. Option C, flower budding. Option C is flower bud differentiation. Option D, fruit development. Option D is the fruit development. And in other column, kinin followed by auxin, auxin followed by kinin, auxin plus gibberellin, auxin plus carbohydrate. A should be 3. If you know 1 also, you can answer the question. Trust me. If you know 1 also, you can answer the question. But it is not applicable all the time. But I am talking about this question. I think option D you can't see. Option D, A will be 1, B will be 4, and C will be 3, and D will be 2. I don't know. Everyone has to give the right answer. Okay. I never ever agree if you give the wrong answer. Use your brain. Slowly, Ravi. Don't be hurry, please. Ravi said C, Karan said C. Anyone else? It's a simple question. It's a simple question. Mahesh also said C. I am requesting all of you, don't make it too complicated. Make it simple. Always make it simple. Don't think too much. When you think too much, you will end up with wrong answer. Always make it simple. Understand what exactly the question being asked. Option C. Okay. Other girls. Shivani, what will be the answer? Archana. Okay, Jagruti, what will be the answer? Okay, friends, it is very easy to answer, right? The moment you see the root differentiation, you no need to think. Root differentiation means what? Auxin. Okay, see, in the tomorrow class we are going to study the gibberellic acid, right? Gibberellic acid is also involved in the process of elongation actually. So which means it is also involved in the root differentiation. If you come to know this, you can easily come to a conclusion. Easily. Okay. And anyway, option 2, if you look into this, auxins followed by the kinins, they will be required for vegetative bud development. And friends, don't get confused. Kinins function is what? Kinins followed by the auxin, they will be required for the flower budding. Don't get confused, friends. And fruit development, you know, right? Friends, if there is a development of the fruit, what do we require, friends? For the development of the fruit, we always require the sugars. Correct, na? So then sugar plus hormone. Correct. Okay. Friends, I hope everyone answered it correctly. Okay. Is it clear, everyone, so far? Okay. So friends, so that's all for today. Tomorrow target is little bigger because tomorrow we will be conducting 7 to 8:30 p.m. My ultimate goal is 7 to 8 p.m. Every student has to study this particular, what we can say, that 4 hormones. We have to complete gibberellic acid, we have to complete ethylene, we have to complete the cytokinin, and we have to complete the what is that other hormone, abscisic acid. So what we do is for every hormone we will keep 15 minutes. Every hormone we will keep 15 minutes. In 15 minutes we will read all 4 hormones. Then what we will discuss is, then we will discuss about the 30 minutes questions. First I will solve PYQs. If you are answering 70% of PYQs correctly, your preparation is going extraordinary. Okay. If you are not answering less than 70%, if you are answering less than 70%, you have to change the way of preparation. Okay. And friends, whatever the extra information that I have discussed about the auxin, especially the inhibitors, please remember that without fail. Okay. Okay. Mauli, chemiosmotic theory. Okay. Okay. Mauli, I will explain in 5 minutes. Just follow correctly. Chemiosmotic theory is actually explaining about the polar auxin transport. What did I tell you about the polar auxin transport? The auxin is moving in a unidirectional from shoot to the root. Look into this. This is the cell number 1. This is the cell number 2. This is the cell number 3. Auxin can exhibited in how many forms in the plant body? Auxin exhibited in the IAAH form, uncharged form. Auxin exhibited in the IAA minus form. Everyone remember, whenever a auxin which is in the uncharged form, what form? Uncharged form. Uncharged form can enter easily, friends. All the students look into the diagram. IAAH can easily enter into the cytosol. See here. Is there any requirement of any kind of transporter is required? No. IAAH directly enter. But if this IAA minus wants to enter, it is a charged form. You know very well. Does the plasma membrane yellow the charged compounds? No. None of the ion never cross the plasma membrane. Yes or no? So what is happening? The charged form of IAA minus, what is happening to this? IAA minus, friends, it is entering via carrier. It is entering via what? Carrier. What is that particular carrier, friends? That particular carrier only we call it as the influx carrier. So all the students follow me. Now Simon, look into the point. IAAH directly entered. IAA minus entered via influx carrier. Now you know what this IAAH will do? IAAH lose the proton because in the cytosol, what is the pH? pH is 7. So in the 7 pH, what happened? It lose the proton. IAH converted once again into what? IAH converted once again into IAA minus. Then the IAA minus, can it go on its own? Because it can't cross the plasma membrane. So it obviously require what, friends? It obviously require the efflux carriers. That efflux carriers only we can call pin proteins. So where the efflux carriers are present? Exactly here, exactly on the basal position. So IAA minus also come like this. Suppose example, if efflux carriers are located like this, auxin also move like that. So what my statement to all of you is, how auxin has to be transported, it is depending on the efflux carriers. 99% of the cases how the efflux carriers are present at the basal position. That is the reason from the shoot auxin will always travels in a single direction. This is called as polar transport. I hope everyone understand. I strongly believe from my side every student is really good with the hormone auxin. Still, if anyone find any kind of a doubt, feel free to ask me anytime. Tomorrow target is finishing the 4 hormones. 15 minutes we will discuss, we will study every hormone, because in the tomorrow 2 classes I'll finish all the hormones. Then 30 minutes we will do the question discussion of especially previous. Okay? That is the plan. Okay, see friends, reading one day is not the matter. You have to read 45 to 50 days consistently. Then you will get enough of confidence to crack that particular question. Okay, I'll help you in every part. Okay, thank you friends. That's all for now. I hope the discussion is useful. I will see you tomorrow sharp 5:45 AM. Thank you all. Work very hard friends. Thank you so much. Thank you all. I appreciate all your patience. Thank you.